Now, without further ado, I am pleased to introduce our first speaker. Dr. Mark Lindsay is co-director of the Myhre Syndrome Clinic at Massachusetts General Hospital in Boston and a distinguished cardiologist known for his expertise in cardiovascular and connective tissue diseases. Dr. Lindsay is leading the Myhre Syndrome mouse model research and in clinic provides ongoing care for children and adult patients with genetically triggered vascular disease. Dr. Lindsay, thank you for being here. We know many Myhre families are excited to hear your research updates. So over to you.
Yeah, not at all. I just tried to share and it said it's disabled. So.
Oh, Amanda, who is our technical director, will rectify that immediately.
Yeah. There it goes. Okay.
Great, thank you.
Is that visible?
Yes, thank you.
Yeah, hi, thanks so much. Happy to give an update on this, in our work, both trying to look at some of the clinical features of patients with Myhre syndrome and in terms of their cardiovascular conditions, and then also an update on the very perplexing mouse model we built and give you where we are with it. So just disclosures, I have research funding from a company called Angio Biotherapeutics that's not related to Myhre syndrome. So, we know Myhre syndrome is caused by these two recurrent mutations in SMAD4. And these happen, at the end of the gene. One is in an arginine 496, and the other is isoleucine 500. We're just starting to sort of look and see if we can compare folks with one or the other, and I'll present a little bit of data about that, but this helps us kind of understand where the condition comes from, but we don't really fully understand what these gene changes do, even to the SMAD4 protein. But we know a lot about SMAD4. It's a fairly famous molecule in biology. And what it does basically is it's involved in what's called TGF-beta's superfamily signaling. The way this works is that TGF-beta ligands, which you can think of as sort of their hormones, they go to the cell surface and they bind to this receptor complex, the TGF-BR1 and TGF-BR2 complex. And that complex, when activated, it's what's called a kinase. It transfers phosphate groups to other proteins. And so it phosphorylates a protein called SMAD3, or actually SMAD2 or 3. And then there are, of course, in the superfamily, there are other SMADs like SMAD1 and 5 that are phosphorylated by different receptors. But in the case of SMAD4 is what's known as a co-SMAD. It binds to these proteins like SMAD2 or 3 and helps them move into the nucleus. And within the nucleus, they direct gene transcription, which is sort of how a cell changes its environment and behavior. So basically, this is how you get a hormone to come from the outside and affect things on the inside of the cell. But as I alluded to, the superfamily is bigger than just classical TGF-beta. There's other ligands like bone morphogenic proteins, or BMPs, that help build bone and other calcium-containing structures, and activins, which are a very cardiovascularly active family as well. And SMAD4 is involved in transducing all of these signals. So when we think about how SMAD4 function is disrupted in Myhre syndrome, it could be through any of these or all of them, and we're trying to tease that out. So I want to just give a quick update. We're going through our clinical experience at MGH, and we're doing what's called phenotyping, or trying to really in detail describe the features in the cardiovascular system. We know that, from some of our early observations, we know that the aorta, and actually all of the large, what we call conductance vessels, are somewhat diminutive, and especially in this region in the abdomen. And this is in contrast to other TGF-beta associated conditions like Loeys-Dietz syndrome, in which their arteries are enlarged. It's kind of so we've been looking at the patients who have come through our clinic and trying to better define the aorta. And let me just explain a little bit to help you understand these observations. So the way, because when we examine children, we can't use an absolute measurement of any sort of anatomy, because we have to index any structure to the size of the child, because children are small, and then children are medium sized, and then they're adults. So what we use is this thing called a Z-score, and a Z-score, you can think of it as just how far away you are from the middle of the population. So a Z-score of 0 is basically dead center in the population. That's so, and then a positive score is your whatever anatomic structure that is bigger than the average, and a negative score is that it's smaller than average. So these plots represent different regions of the aorta, and the solid black line that's horizontal is where the average Myhre syndrome patient falls in this Z-score. And you can see that all of the regions shown here, this is the ascending aorta, which has 4 regions that we measure in medicine. They're all smaller than they should be. And it's so, one thing is we've been staring at the middle of the abdomen and looking at this narrowing, but it's really a more generalized finding. In fact, when we look down the descending aorta, we see the same thing. We see that there's aortic hypoplasia across all these regions, and really across all the way down into the arteries in the legs. What this tells us is that the vascular system, the large vessel vascular system, is somewhat undersized. There are very few human conditions like this. The only other one I know analogous is called Williams syndrome. It also affects arteries and makes them somewhat smaller. And when we look across these populations, we also see that something probably not too surprising to parents, but that the boys are a little more affected than the girls on average across the aorta. So, we're probably seeing, and this is fairly common in cardiovascular disease writ large, is that men, for whatever reason, tend to have worse cardiovascular disease, all things equal. We see this in Marfan syndrome, we see this in Loeys-Dietz syndrome, and we see it in just general cardiovascular disease like coronary artery disease. As far as the 2 gene changes go, the arginine 496 and the isoleucine 500, it looks like, at least from this feature, this phenotype, that the isoleucine 500 is a little more severe in terms of the aortic hypoplasia, a little smaller aortas as measured by us. So that's what we're doing. We're expanding to figure a little bit more about the characteristics of those arteries and why they're small. But one of the results is that we end up seeing a systolic high blood pressure, a systolic hypertension, which makes a lot of sense because the heart has to pump into a smaller vasculature. We're also right now tabulating what's called the diastology, or the way the heart relaxes. I don't have that ready to show. We just got all the data, and we have a statistician who's going to put that together. And so I look forward to getting this all out to the community in the next year. I just wanted to mention we are doing that. So let me move on to the mouse model. We have a lot of experience in medicine, and especially in rare disease, using mouse models to model different kinds of rare disease. This is from work I did a long time ago, comparing arterial systems in mice. So these are little mouse hearts, and this is their aorta. This is a model of Marfan syndrome, or an FBN1 mutation. The other one is a model, TGFB2, is a model of Loeys-Dietz syndrome. And, we know that mice, they don't recapitulate every human disease very well. Cancer, for instance, is an example of a disease where we constantly cure cancer in mice, and then when it moves to people, it's more difficult. And that's probably because the mice don't really reflect the complexity of cancer biology. But mice tend to do pretty well in arterial problems. So we designed, with the help of the foundation, this mouse. We decided to go for the isoleucine mutation. The reason for that is because I had the sense that it was a little more severe. And the worst thing that you want after you build a mouse is that it doesn't have a phenotype. It just looks like a regular mouse, which is a huge disappointment, but sometimes happens. This is a special kind of design that it's called a knock-in, where we can basically turn on the mutation. And so let me just give you just a little understanding of how we do that. For those of you who aren't familiar with this technology, there's an enzyme called Cre recombinase. We can express this inside animals or inside cells or wherever we want. What it does is, it's literally what it's called. It's a recombinase. It can recognize two sites in a DNA strand. They happen to be called LOXP sites. And when it recognizes those, it actually loops out that DNA and removes it from the stretch of DNA that it's in, and it leaves one LOXP site there, and it puts the other out in this circular piece of DNA. Well, so how is that useful? It's useful because, that's the Cre, and that's what you end up with. It's useful because in animal models, we can express Cre, direct it into the places we want to understand the mutation to be turned on, or temporally, meaning if we want to turn it on later in the lifespan of the animal, we can do that. So how does this work for our mouse that we're interested in? What happens is that this cassette, which has the mutation, when the Cre fluxes out this exon 12 actually, it advances the mutation, and it just incorporates the new mutation. And so you can get, in what we call heterozygosity, one copy to turn on the mutation, just like what we have in Myhre syndrome. And so then you cross that mouse to a mouse that expresses the Cre recombinase, and their children, their babies, will turn on the mutation. And so we first wanted to understand the context of Myhre syndrome. So we use what's called a CMV Cre, and that is an animal that expresses Cre in every cell. So it's just like a person with Myhre syndrome, they're going to express the mutation in every cell. That's where you kind of want to start from if you're studying a congenital disorder. So first of all, what we got was they are phenotypically affected. They are a little smaller than the average mouse. So this is a mouse with Myhre syndrome on the right, and that's its littermate or sibling, if you want to think about it that way, on the left. They are a little smaller, just like folks with Myhre syndrome are a little smaller. So we're happy to see that. And we've now gone through and looked in different organ systems and trying to get what we sort of call a survey of these animals to see what phenotypes they express, and do they look like patients. And so I was happy to see that there are smaller aortas in the Myhre syndrome mice, and we're trying to quantitate what that is and what that means. The other thing we see in Myhre syndrome patients is, fortunately in a smaller subset, but in some patients, something called restrictive cardiomyopathy, which is an inability of the heart to relax. The mice have evidence of this, both at the histologic level, which is what you're looking at. This is what the you probably haven't seen pictures like this, but this is what the heart looks like if you cut it like a loaf of bread. It has two chambers, and in the Myhre mice, the chamber is pretty muscle bound, so it's probably a little smaller. But physiologically, we see that there is evidence of diastolic dysfunction. So this is a map of the mitral inflow, which allows us to understand how the diastolic function of the heart works, or basically how well the heart relaxes. And we see in the Myhre mice, there's a different E/A ratio, which is what we use actually in cardiology in clinic to look at advanced diastolic dysfunction. So we're happy to see that the animals also had that. I guess that's okay. Flipped ahead. We've also been looking through all of the organs to see are there other features that we see in patients that are expressed in the animal. So we've noted several. One is kidney fibrosis. This is, you can stain it different ways, but in this case, as measured by alpha smooth muscle actin expression, which is upregulated in tissue that's undergoing fibrotic change. And so we've systematically gone through and noted strong phenotypes in most organs. Don't really see anything in the liver. But in most of the organs that we've looked in, we can see differences, and we're trying to map these differences at the moment. One thing I wanted to touch on was our group has looked in the MRI scans from patients. We've noted these white matter changes, and that's a sign, can be a sign of vascular disease in the brain. And so we've taken some special attention to look at the vascular system and the neurovascular system within the brain, and do see some subtle changes. We haven't quantified them yet, but do see basically the arterial system in the brain looks a little bit like the arterial system in other parts of the body, a little small and a little hypertrophied, or a little thicker arteries. This could be from high blood pressure, or it could be intrinsic to the arteries, and that's one thing we're trying to work out now. But an important aspect of how the neurovasculature works, which is different than the way the rest of the vasculature works, is that the structure of small arterioles in the brain is different. And it's different because it has this thing called the blood-brain barrier, which basically protects the brain itself from both from the effects of vascular disease like high blood pressure, atherosclerosis, but also from toxins and things that you don't want your neurons being exposed to. And we noted in the Myhre mice basically an activation of the astrocytes, and this is a common sign that when the vasculature isn't performing as well as it should. And so this is something we think is hugely important from a patient standpoint, and so we're going to be drilling down into this. And so just a summary slide, it's preliminary that there is something different about the neurovasculature, and that we're going to be looking at that extensively. So this is good, right? The whole point of making this animal is that we want to start trialing things, right? We want to start using drugs and pharmacologic agents, and we want to think about gene therapeutics and like how we can do it. And so I think it's great news that we have an animal that's that's affected in multiple organ systems, and it's going to tell us a lot. So we were geared up, and we started putting together more and more cages and to try to really expand the colony. And we ran into a problem, and that was when we, you put two mice together to breed. They have a litter that's about 8 mice on average. That's how many babies an average mouse pregnancy has. And what we would find And this particular breeding strategy, I should tell you, creates basically a 1-to-1 ratio of the animals with the mutation turned on and the animals without the mutation turned on. And so what we found was that when we actually don't check the genotype until about a month, because it's more convenient for just technical purposes, what we found is that we were only finding females, or at least a heavy skew towards females, about 80% to 20% when the expected ratio is 50-50. And not only were we finding a very big skew of females, which basically suggests male mortality early. But when we went back and started watching litters much more carefully, we found that there was a ton of basically perinatal lethality. What that means is death within the first several days of birth. That's classically in mouse biology often a congenital heart disease effect, because mice, unlike humans, don't get surgical treatment when they have congenital heart disease and they pass away. But I hand examined a whole bunch of hearts, and they don't have congenital heart disease. But I was fortunate, and I think Hongmei is online today, but to have a colleague, Hongmei Mu, who's here in our cutaneous biology division and is really interested in how the epithelium works. And she had previously been involved in SMAD4 biology from that standpoint. And she had noted that we had given her tissues to analyze and had noted signs of recurrent pneumonia in some of the older animals. And so then went back and looked at these very young mice, so day 7 of life, and found evidence of inflammation already in the animals. And then I think in a really impressive observation, she looked into the nasal cavities of the animals within the first week of life and found a amazing, basically that the nasal cavities are completely filled with inflammatory infiltrate. So a lot of mammals, including humans and mice, when they're babies, are called obligate nasal breathers. You may not have heard this before, but basically, if you can't breathe through your nose, you can't nurse. And if you can't nurse, you can't grow. And so we think that these mice are basically can't breathe, you can't nurse, and therefore dying early. And that this is because of this phenotype. What's really interesting, Hongmei is going through now and has made some really interesting observations that this isn't by accident. It's actually SMAD4 is important in determining the cell types in the nasal passages. And she's now going through, this is not her data, this is just an example of how we look at the nasal epithelium. But there's a difference in the cells, and the cells aren't performing well because of this SMAD4 gene change. And this really complements the observations in our clinical experience, where a lot of kids and even adults have had recurrent respiratory infections, asthma that's not responsive to bronchodilators, recurrent ear infections, and sinusitis. So I think, we have there may be a reason that patients with Myhre syndrome are more susceptible. So I think we're learning a lot, not exactly the lessons we thought we would be. When you read an academic paper, the way they're written is sort of, we made this mouse, we tested this drug, and here it works or it doesn't. But actually, there's a lot of trial and error in this kind of biology. And I think we're excited that the animals are, I think, pretty faithfully recapitulating a lot of the features of the condition. Makes them a little harder to work with, but there are technical ways around that. So, I think that the cardiovascular disease is going to be studyable. I think we're going to get some neat insights about the renal and neurovascular systems. And I think we're going to, with Hong Mei's help, really understand better the respiratory epithelium. From a standpoint of using the animals, we can actually, because of the way we designed the mouse, we can actually turn on the mutation a little bit later, like at 2 weeks of age or 4 weeks of age, so that we can get around that perinatal lethality. And we're doing that now, but it's the kind of thing you have to learn experientially. So I just wanted to thank the foundation and everybody online for being here and supporting our work. Happy to take questions.
Yeah, thank you, Mark. We've had some questions come in that we will go through now. So the first question, this relates to earlier on when you mentioned that the constriction seems to be throughout the aorta and funnel all the way down. So does the constriction change with age, or is it static?
I think so far our observation is that it's pretty static. So there's one sort of exception to that, and that is not only people with Myhre syndrome, but spontaneously children can have what's called coarctation of the aorta. And that happens most commonly at a place right near the heart, but on the back side of your aorta, on the aorta, just as it starts coming down the back. That happens shortly after birth, typically, and it's surgically fixed. It's an obligate surgical lesion. That can re-close, it can tighten up again later in life. But that's a feature of everybody that has that congenital heart malformation, not just patients with Myhre syndrome. The generalized hypoplasia of the aorta that's everywhere does not seem to get worse over time. It seems to be fairly static, meaning the aorta is just undersized through growth, and that's probably putting a burden on the heart and a burden on other organs, because it's not as big as it should be, and so the resulting hemodynamic effects are pressure related, and so the body has to deal with higher shear stress and higher blood pressure.
And does that therefore then correlate that as children grow older, if they haven't had congenital heart problems, but they might typically see high blood pressure start to come in, is that because of the stress as they're getting bigger, but the aorta is remaining, as you said, on that smaller side?
Yeah, I think it's gonna turn out to be quite complex, and it's going to turn out to be a relationship between the arterial vasculature and the kidneys, which are the main organ that control your blood pressure. But I think the answer to your question is yes. Certainly, having an undersized vasculature does it basically, your blood pressure is those two numbers, the systolic, which is the top, and the diastolic, which is the bottom, and what that represents is each heartbeat sends a packet of blood, and the pressure goes up and then it goes down, and it goes up and then it goes down. And if your arterial system is undersized or stiff or both, basically it pushes that upper number up, because blood is just coming out at higher pressure, and there's less room for it.
Okay.
So that's what we see. That's what we see in patients is systolic, generally, especially in the kids as they get into adolescence, is isolated systolic hypertension.
Okay. We've just had another question come in, and this is related before we move on to a kidney question. But what symptoms should we be concerned about regarding restrictive cardiomyopathy? So are there screening tools for our doctors to diagnose?
Yeah. I mean, this is one of the reasons we recommend regular follow-up with a pediatric cardiologist, even in the absence of symptomatology, is because the echocardiogram or the ultrasound can help detect some aspects of restriction. It's not perfect though, and so sometimes when people are having symptoms, we have to do further investigation, sometimes even a catheterization, to understand how the pressures in the heart are working. So in patients with restrictive cardiomyopathy, there's high atrial pressure in the heart. The symptoms of restrictive cardiomyopathy are basically fatigue, inability to do exercise at this from what you used to be able to do. The problem, though, is that it's that overlaps with a lot of other causes of fatigue, and patients with Myhre syndrome can have fatigue because of things like claudication or pain in the legs with exercise. So it's not simple to tease out, and that's why we want people to have a relationship with a cardiologist, even if they're feeling okay.
Okay, great, thank you. Kidney fibrosis, that was an interesting finding. So how would that present for symptoms, and what do we all need to look out for in that sense?
So that's something that's probably already being looked out for those who have high blood pressure. We get them, especially kids, we get them with a nephrologist, because in children, the number one doctors who take care of high blood pressure are not cardiologists. They're actually nephrologists or kidney doctors. But I don't know. I mean, we don't have, that's not a strong phenotype in the patients. Like severe kidney, fibrosis has been seen in the patients, but not, I don't think it's common. I don't know if it's going to end up being mostly a mouse thing. Sometimes the mice can have a phenotype because they're a little different than people, but it can still be used to understand whether our treatments respond. So I don't want to give the impression that it's a one-to-one correlation.
Okay. We've had a comment here. Tell us something really positive that's come out of what you've been doing.
Yeah, I think it's very positive to have an animal model that we can start to trial therapies. I know it. This stuff is slow. I worked in a Marfan syndrome animal lab, and it was years from when we created the mouse to when we started scaling up and doing drug trials. But now we have several pharmacologics that we know work in Marfan syndrome, and I think not only that, but this mouse is going to allow us to try some more advanced therapies like gene therapies. And so I think, that was one of my fears, was showing all this gloom of histology and stuff, but it's not. It really, like, you have to understand what's going on before you can fix it, and you have to be able to model it not just in people, especially in ultra rare disease. We're going to get information from patients in ultra rare disease, but we're not going to be able to do a bunch of clinical trials in Myhre. I mean, we want to do them, but there's a limited number of patients, and so we need to have other systems to trial things. So don't give me the impression, or I don't want to give the impression that that it's bad news. Actually, this is how we advance and how we find things.
Lovely, thank you for clarifying that. Another question in: any progress on mouse cell line establishment?
We have some that have just been growing, but if somebody wants them, just send me an email, right? As soon as we have a line out. I'd love to be able to make ES cells or embryonic stem cells from mice, but I don't really have funding for that. But if somebody else does, I'm happy to send them mouse cells, and they can do it themselves.
Great. And that's researchers and scientists. You all can't have a Myhre mouse. Sorry about that, everyone.
Researchers are welcome to reach out, and we can share the cells or whatever.
Okay, lovely, thank you. Another question here: I noticed the heterogeneity of weight and survival. Do you think there could be some other factors contributing to this heterogeneity?
Yeah. So that's always a huge question in animal research. Is you have these animals that are inbred, they have the exact same genes, they're living in the same cages, but you get curves that show heterogeneity. And we always end up spending a lot of time thinking about this. But basically what it represents is that the biology starts from a set point, but there are things that happen in the environment that we basically can't quantify. But that the disease will progress faster in one animal than another, even with the exact same genes. I mean, literally the exact same. These are clones. And so we see this in all animal models, and we see it in people, identical twins. So it's just the nature of entropy and the way disease progresses. But there are definitely factors, absolutely. They are very hard sometimes to get a handle on.
Okay. Thank you very much. I'm conscious of time. If you have other questions for Mark on that presentation, please pop them in the chat and we will endeavour to get those answered. But Mark, one question that did come up is that a lot of the data you've discussed in here, is this going to be published? Does it need to remain confidential on here? If people wanted to look at the slides or share the slides, is that something that's possible, or would you prefer to wait until you have this published?
No, it's all right. I just made it public by showing it to you guys. So.
Yeah, so we were able to share.
It's in the public domain already. Yeah.
Lovely.
It's fine. We share. I want researchers to see it and modify their own experiments. I know that there's other animal models, in Europe, and I hope they can use this experience, or we can use theirs. And it's just. But yeah, you can do whatever you want with the slides, but I would, if you want an interpretation, feel free to email me. So I don't want people over interpreting a mouse.
Yep.
To make it to be too much towards people. Mice can help inform people, but they're not people.
Great. Lovely. Understood.